The rising concentration of atmospheric carbon dioxide, or CO₂, is one of the most prominent environmental changes associated with climate change. Since the Industrial Revolution, atmospheric CO₂ levels have increased significantly, from around 280 parts per million to more than 420 parts per million today, with projections indicating further increases throughout the 21st century. This change has significant implications for plant physiology, agricultural productivity, and the nutritional quality of crops. As photosynthetic organisms, plants directly respond to elevated CO₂ levels, leading to alterations in growth patterns, water use efficiency, and biochemical composition. However, these physiological responses are complex, varying across plant species, environmental conditions, and agricultural practices. While elevated CO₂ may stimulate plant growth, it can also lead to declines in grain quality, particularly in essential nutrients such as proteins, minerals, and vitamins. This documentary explores the intricate influence of elevated CO₂ on plant physiology and grain quality, supported by extensive evidence, scientific studies, and global research efforts. The historical increase in atmospheric carbon dioxide concentrations has been meticulously documented through ice core samples, atmospheric monitoring, and remote sensing technologies. Pre-industrial carbon dioxide levels remained relatively stable for thousands of years until human activities, particularly fossil fuel combustion and deforestation, significantly increased emissions. In 1958, Charles David Keeling initiated the systematic measurement of atmospheric carbon dioxide at the Mauna Loa Observatory in Hawaii, revealing a consistent upward trend known as the Keeling curve. Data from the World Meteorological Organization indicates that atmospheric carbon dioxide reached 419 parts per million in 2023, with the Intergovernmental Panel on Climate Change projecting concentrations between 550 and 700 parts per million by the end of the century under high emission scenarios. The primary mechanism through which elevated carbon dioxide influences plant physiology is photosynthesis, the biochemical process through which plants convert light energy into chemical energy. Photosynthesis involves two pathways, C3 and C4 photosynthesis. C3 plants, which include rice, wheat, and soybeans, rely on ribulose 1,5-bisphosphate carboxylase oxygenase, or RuBisCO, to fix atmospheric carbon dioxide during photosynthesis. This enzyme, however, also catalyzes oxygenation, reducing photosynthetic efficiency. Elevated carbon dioxide levels mitigate this limitation by increasing substrate availability for carboxylation, thereby enhancing photosynthesis and growth. Numerous free air carbon dioxide enrichment experiments, often referred to as FACE experiments, have demonstrated that C3 plants often experience significant increases in biomass under elevated carbon dioxide conditions. For example, the RiceFACE experiment in Japan found that elevated CO₂ at about 580 parts per million increased rice biomass by approximately 15 to 20%, depending on nitrogen availability and cultivar differences. In contrast, C4 plants such as maize and sorghum utilize phosphenylpyruvate carboxylase, or PEPC, to initially fix CO₂, which reduces photorespiration. As a result, C4 plants exhibit smaller photosynthetic gains under elevated CO₂ conditions compared to C3 species. Research conducted at the University of Illinois demonstrated that maize grown under elevated CO₂ exhibited minimal increases in biomass unless accompanied by other factors, such as higher temperatures or water stress. Elevated carbon dioxide also influences plant-water relations primarily through changes in stomatal conductance. Stomata are microscopic pores on leaf surfaces that regulate gas exchange, including the uptake of carbon dioxide and the loss of water vapor through transpiration. Under elevated carbon dioxide conditions, many plant species exhibit partial stomatal closure, which reduces transpiration rates and improves water use efficiency. A meta-analysis conducted by Medlin et al. in 2011 across 90 plant species found that elevated carbon dioxide increased water use efficiency by an average of 20 to 40%. The increased availability of carbon dioxide often alters carbon allocation patterns within plants, influencing biomass distribution across leaves, stems, roots, and reproductive organs. Elevated carbon dioxide typically promotes greater carbon allocation to below-ground tissues, enhancing root growth and rhizosphere interactions. Root system expansion can improve nutrient uptake, particularly for immobile nutrients such as phosphorus. Research conducted by Ainsworth and Long in 2005 showed that wheat plants grown under elevated CO₂ allocated up to 30% more carbon to roots compared to ambient conditions. In terms of yield, elevated CO₂ has consistently been associated with increased grain production in C3 crops. FACE experiments in China demonstrated that elevated CO₂ at 550 parts per million increased rice yields by 12 to 15%, with similar results observed in wheat and soybean. Despite the positive effects of elevated CO₂ on biomass production and yield, concerns regarding grain quality have gained increasing attention in recent years. Numerous studies have reported nutrient dilution effects, wherein the concentrations of essential nutrients in grains decrease under elevated CO₂ conditions. Proteins, minerals like iron and zinc, and vitamins often decline in cereal grains grown under high CO₂ levels. For instance, a global meta-analysis by Myers et al. In 2014 examined data from FACE experiments across multiple continents. They reported that wheat grown under elevated CO₂ had 9.3% lower protein content, 5.1% lower zinc concentration, and 5.2% lower iron concentration compared to ambient conditions. The protein dilution effect is primarily attributed to increased carbohydrate accumulation without a corresponding rise in nitrogen uptake, resulting in lower protein to carbohydrate ratios. One major factor is the disproportionate stimulation of carbohydrate synthesis relative to nutrient uptake. Elevated CO₂ enhances photosynthetic carbon fixation, leading to increased starch accumulation in grains. However, nutrient uptake processes such as nitrate assimilation and micronutrient transport do not increase proportionately. Studies conducted at the University of Western Australia found that wheat plants grown under elevated carbon dioxide exhibited impaired nitrate assimilation, as nitrogen metabolism is regulated by processes that are less responsive to carbon dioxide enrichment. The decline in grain nutrient content has significant implications for global food security and human health. Cereals such as rice, wheat, and maize provide the majority of dietary calories and nutrients for billions of people worldwide. Reductions in protein, iron, and zinc content under elevated carbon dioxide conditions may exacerbate malnutrition, particularly in developing regions where dietary diversity is limited. A study published in Nature Climate Change projected that elevated carbon dioxide levels by 2050 could place an additional 150 million people at risk of zinc deficiency and 122 million at risk of protein deficiency. Furthermore, micronutrient deficiencies, often termed hidden hunger, can have severe health consequences, including impaired cognitive development, weakened immune function, and increased morbidity and mortality rates. Iron deficiency, for instance, is already the most prevalent nutritional disorder globally, affecting over 2 billion people. Elevated CO₂ could worsen this crisis by reducing the iron content of staple grains, with potential intergenerational impacts on maternal and child health. The physiological and nutritional responses of plants to elevated CO₂ are further modulated by interactions with other environmental factors such as temperature, water availability, and nutrient supply. Rising global temperatures may synergistically or antagonistically interact with elevated carbon dioxide, depending on crop species and growing conditions. For example, heat stress can exacerbate nutrient dilution by accelerating grain filling and reducing the time available for nutrient accumulation. Water availability also plays a critical role in modulating plant responses to elevated carbon dioxide. While increased water use efficiency can benefit crops in semi-arid regions, prolonged droughts can limit nutrient uptake and compromise yield gains. Nutrient availability, particularly nitrogen, is another crucial determinant of plant responses to elevated carbon dioxide. Numerous studies have shown that nitrogen fertilization can partially mitigate nutrient dilution effects by supporting enhanced nitrogen assimilation and protein synthesis. Addressing the challenges posed by elevated carbon dioxide requires the development and implementation of adaptive agricultural strategies. One promising approach is the breeding and genetic modification of crop varieties with enhanced nutrient uptake, nitrogen use efficiency, and grain quality. For instance, researchers at the International Rice Research Institute have developed rice lines with increased iron and zinc content through biofortification techniques, mitigating nutrient dilution effects under elevated carbon dioxide conditions. Improved agronomic practices, such as optimized fertilization regimes, crop rotation, and soil health management, can also support plant nutrient acquisition. Precision agriculture technologies, including remote sensing and soil nutrient mapping, enable more efficient resource allocation and adaptive responses to changing environmental conditions. Additionally, sustainable soil management practices, such as conservation tillage and cover cropping, enhance soil organic matter, microbial diversity, and nutrient availability. The physiological and nutritional impacts of elevated CO₂ extend beyond agricultural production, influencing global food security, public health, and socioeconomic development. Policymakers must prioritize research and development initiatives that address the nutritional impacts of climate change with particular attention to vulnerable populations in low-income regions. International collaborations, such as the Global Research Alliance on Agricultural Greenhouse Gases, can facilitate knowledge exchange, technology transfer, and the development of climate-resilient agricultural systems. Furthermore, reducing greenhouse gas emissions through climate mitigation policies remains fundamental to limiting atmospheric CO₂ increases and minimizing associated impacts on plant physiology and grain quality. The influence of elevated CO₂ on plant physiology and grain quality is a multifaceted and dynamic phenomenon with significant implications for global agriculture and food security. While increased CO₂ can stimulate photosynthesis, growth, and water use efficiency, it simultaneously compromises grain quality by reducing essential nutrients such as proteins, minerals, and vitamins. Addressing these challenges requires interdisciplinary research efforts, innovative agricultural practices, and proactive policy interventions to safeguard global food and nutritional security in a changing climate. As atmospheric CO₂ concentrations continue to rise, understanding and mitigating these impacts remains a critical scientific, agricultural, and societal imperative.